Hello everyone, my name is Michael Steger, and today I'll be presenting on polyelectrolyte complexes as a biomaterial system for bony bar prevention in physeal injuries. So if we direct our attention to figure 1, you can see that the growth plate, otherwise known as the physis, is this cartilage region located at the end of long bones. When the growth plate is fractured, you can get the formation of a bony bar, which can cause angular deformities or growth arrest. Our method for treatment is using polyelectrolyte complexes, or PECs, which are depicted here in figure 2. This top monomer, alginate, has negative charges, and this bottom monomer, chitosan, has positive charges. These two polymers can form electrostatic interactions with each other to form a PEC hydrogel system. The main objectives of this study were to fabricate these PECs with varying biopolymer composition and mechanical characteristics. Then we wanted to grow human mesenchymal stem cells throughout the PEC scaffolds and study the stem cell differentiation and proliferation as a function of the scaffold composition and mechanics. So looking at figure 3, we can see that we have precise control over the mechanics of the hydrogel scaffolds simply by varying the biopolymer composition within them, ranging from about 15 kPa storage modulus at the stiffest scaffold down to about 2.5 kPa storage modulus for the least stiff scaffold. Figure 4 shows the entire oscillatory strain sweep at a constant 1 Hz amplitude. Next, we had to measure stem cell differentiation. And to do this, we measured alkaline phosphatase expression, or ALP, which is an early osteogenic marker, and glycosaminoglycan deposition, or GAGs, which are an indication of cells' ability to lay down their cartilage extracellular matrix. As we can see in figure 6, if we include hyaluronic acid within our scaffolds, it decreases the alkaline phosphatase expression in both cases. We also see that our most stiff scaffold, gel 3, and our least stiff scaffold, gel 1, have the highest alkaline phosphatase activity, meaning that the ideal mechanics for the scaffold should be somewhere in between these two, so gels 2 or gel 4. From figure 7, we can see that if we include calcium as a crosslinker in our scaffolds, it actually has a deleterious effect on the glycosaminoglycan deposition, meaning that gels 1 or gels 2 would be the most ideal in this case. The next steps for this project are to include these scaffolds in an in vivo rat injury model, which are shown in figure 5 and figure 8. In this model, a drill hole is done through the growth plate, and then the biomaterial scaffold is placed within it. As we can see in figure 8A, this is the untreated, and in figures B, C, and D, this is where the biomaterial scaffold has been inserted, and there is less bony bar formation. Additionally, some more next steps include releasing therapeutic small interfering RNA from the PEC scaffolds targeting the P38 MAPK pathway. That concludes my presentation. Thank you all for listening.